Okay, so how do things get in and out of this? So you've got this plasma membrane here, right? It's our phospholipid bilayer. So I'm just drawing it right here. This is the phospholipid bilayer, okay? It's our cell membrane. So the key concept is that simple diffusion is going to allow molecules to go through, right? They're going to pass directly through the hydrophobic region. So what is going to be allowed through directly? Okay, that's called simple diffusion. Simple diffusion. It's going straight through. Does not require a protein, right? No protein required. Required. So what goes through? You do need to know they could easily go in or out. The things that go in or out need to be non-polar. So something that's going to go into the cell is oxygen. There's a lot more oxygen outside of the cell than there is in it. So it's going to move into the cell. Another molecule that can go straight through is carbon dioxide. So I want you to know that. So carbon dioxide gas, it's produced in the cell. So it builds up in there. And it's also relatively non-polar. So there's more inside than outside. So it's going to be moving out. The main thing is everything that goes through needs to be pretty small, can't be a huge molecule, and it needs to be non-polar. So it can't have a big charge on it. Excuse me, it can't have a big charge on it. Okay. Another thing we're going to get to later, I'm just going to put it in here, but another thing that can go in is a steroid. So steroids, if you remember, are lipids. Lipids are non-polar, and so it can go straight through. So this region that it's going through is hydrophobic. So it's only going to allow through non-polar molecules. Only non-polar gets through. Okay. Now, I am going to make one exception because I don't want the exception is water. So it's a little bit of water can get through. But other than that, everything else needs to be non-polar. Okay, so water is when water diffuses across a membrane, it has a special name. It's called osmosis. So osmosis is the diffusion of water across a membrane. And so what's shown here, this is a little bit odd because the water is actually pink. So here's the water. I should just draw it in pink. Weird color for water to be, but here's water. Water is the most common thing here. So all of this is water. And this is showing that water is actually going to move through this semi-permeable membrane here shown in red. And it's going to eventually go to this side. And now you've got more water over here than you do on the left. So this process where the water moves through is called osmosis. All right. And again, this is simple diffusion. Simple diffusion. So diffusion is the direction of the diffusion is always going to go from a high concentration to a low concentration. So in this example, if you have a high concentration of, say, water outside the cell and a low concentration inside, then it's going to go from high to low. Okay. And this is showing simple diffusion. So it's going straight through. So this is simple diffusion. Now notice it does not require a protein. It's just going straight through. And this is going to continue to occur until you have the same number of molecules on the inside as you do on the outside. And then you will not have any net movement, okay? It'll be in homeostasis. Okay, so how many more do I have? Oh, okay, I'm trying to make these 10 minutes, so. Okay, let me go through this one quick. So although water molecules are polar, they go through the membrane through osmosis. So that can be really quick. So water goes through the process, the cell membrane. This is called osmosis. Okay, perfect. Now, the direction we talked about, tonicity. So there's three ways that you can describe the solution surrounding the cell. So here's my cell, okay? And if there's a whole bunch of solutes outside the cell, okay, that is going to be called hypertonic. And if it's hypertonic, the water itself, okay, is going to move out of the cell, okay, because there's actually going to be more water in the cell and less water outside of the cell. So the water is going to move out. Okay. Isotonic is where you have equal concentration of solutes inside the cell as you do outside the cell. So water moves in both directions, but it's in balance. So there's no net movement. Okay. So over here on the left, the water moved out. The cell will actually shrink if it's an animal cell. This one's going to be the same size. And then if it's hypotonic, you've got a cell, you've got a few solutes outside, a whole bunch inside. And water is actually going to go into the cell, and then the cell will expand. Okay. Diffusion rates. So if you have a higher concentration gradient, then you're going to have a higher rate. What that means is, say I just have, 2 or 3 molecules out here and 1 in here, they will move in. But if I happen to have another scenario where I have 100 out here and 1 in here, you're going to get that movement faster. So the higher the concentration gradient, the faster the rate. Also temperature. So this is kinetic energy, right? So it's how fast the molecules are moving. So a higher temperature is a higher speed of molecules. That means they're actually going to just be moving, bumping into each other a lot faster. So they're going to get in quicker. The surface area as well. So say I have one cell that is like this, a sphere, and I have another cell that with the same volume, it's really long and narrow, okay? This one has a higher surface area relative to the volume inside. Okay, this has a lower surface area relative to the volume. So this one is going to be faster. It's a faster rate, and this will be slower. And if you think about, okay, I've got different colors. So I've got this many molecules, right? And here's some here. There's all this area down here below where it can go straight into the cell. So there's tons of places where these molecules can go in, and it's more relative to the volume than up here. This would be actually less. Okay, and that's it for this video.